In the last year, during the ABS virtual meeting, we revealed that Nearctic Neotropical migratory shorebird species join heterospecific flocks in a balanced number to control for interspecific competition, also maintain the flock protective effect in South America. This last manuscript raised some additional questions about interspecific relationships between flocking species. For example, are these species distantly related, and thus do these species have different functional traits that favor their participation in flocks? Here we explored if some traits linked to the fundamental niche of species present phylogenetic signals and explain the formation of flocks. We recorded heterospecific flocks in urban beats from southwestern Brazil, annotating the species richness and species abundance per flock. We used Bloomberg's K to discern if bud mass, bill, and tarsus lengths have phylogenetic signal. The mental test was used to identify a relation between phylogenetic variance, covariance metrics, and frequency of co-participation between species. The GLM was used to find the best model that explained the relation between modular difference in traits between pairs of co-participating species and their frequency of co-participation in flocks. We also used the species strength concept to identify the general overview of the dependence of shorebird species in heterospecific flocks. We recorded 7 species of Nearctic Neotropical migrants in 65 heterospecific flocks. The semi-palmated plover exerts the highest momentary dependence on other species in flocks. As shown by the mental tests, co-participation between pairs of species were not related to their relatedness. Also, as shown by the Bloomberg's, bud mass, bill length, and tarsus length did not explain the structure of phylogenetic tree. GLM analysis showed that co-participation between shorebird species tended to decrease as difference in bud mass and tarsus length increase. Our results discard the influence of relatedness between shorebirds and that the chosen phylogenetic filter and signals could be acting in the formation of heterospecific flocks. Our significant results showing similarity of bud mass and tarsus lengths open a discussion about the influence of biotic mechanisms on the attractiveness of individuals in heterospecific flocks, thus indicating for the first time a constructive effect for this group of birds. For people that are interested in more detailed information, this article was published recently in the Outlined Journal.